So our next speaker is Meghan, and Meghan, yeah, okay, are you gonna come up here? And she's gonna talk to us about genetic and environmental drivers of red oak phenology. Okay. Do you have all the images?
I have like, it's a 3-minute talk, but my gosh, is it high def? Apparently.
One extremely detailed slide.
Super detailed. Just imagine it in your mind's eye. I feel like it may have stalled. Is it good? You got it? Yay! It's that first slide. Great. Okay, so this is a short talk. It's just 3 minutes. So I figured I'd give you a quick overview of sort of what I'm doing for my postdoc fellowship. I'm 2 years into a 3-year NSF PRFB in PGRB Plant Genome Research Program. And I'm studying sort of the genomic and environmental drivers of phenology in red oak, or Quercus rubras, northern red oak. And so the background here is a drone imagery of the Harvard forest. So you have a swamp over there and sort of not swamp over here, and you have all these red oaks in this image. And what sort of prompted this research was many, many years ago in my lab, someone had done all these drone measurements and looking at phenology and found that there was this huge diversity of phenological timing in red oak in this area, this one little area, that the red oaks on this side of the plot would leaf out a month later than the red oaks on this side of the plot. And I was like, oh, that's super interesting. And he found that environmental, microenvironmental parameters only explain about 15% of that variation. I was like, well, is it genetic? And he's like, I don't know, I don't do genetics. So that sort of prompted this whole postdoc that I ended up doing many, many years later. And so phenology is super important. It is a huge predictor of global climate change. Spring now occurs 2 weeks earlier than it did pre-industrial times. That means that not only is carbon being taken up earlier, but the Earth's planet albedo is changing and water fluxes are changing, which has a huge feedback into the global climate system. And so modelers, ecosystem demography modelers care a lot about getting phenology timing right, but they completely ignore the fact that phenology has a huge heritable component to it and that it's strongly locally adapted. So what I'm super interested in doing for my postdoc is sort of parsing apart that variation, modeling out sort of using both genetic and environmental variation to understand what species range shifts might look like if you consider both environmental and genetic variation, and then I'm going to validate that. So again, I'm 2 years in, so I've already completed this, but I have a growth chamber study where it's really hard to set up a common garden of red oak in a 2-year postdoc or 3-year postdoc. So I ended up taking leaf branch cuttings post bud set and put them out into different environmental manipulations in our growth chamber and was able to force bud break. And so I observed bud break in 8 different genotypes sourced from the Arnold Arboretum, anywhere from Alabama all the way up into New Hampshire. So their provenance is from across their entire range. So I'm looking at both genetic and environment across all these different environments. I can come up with the reaction norms. I can then put these reaction norms into this phenofit model. It's a process-based model created by Isabelle Schween in Montpellier, France. And the beauty of process-based models is we're entering this non-analog climate future, as someone mentioned earlier. So we can create, sort of these statistical models based on sort of what we see on the landscape today and how it matches to climate. But climate is changing in ways that we don't actually have a map to make today. So the benefit of process-based models is that you sort of understand how plants work and can use that sort of to better understand sort of how they might change with climate. So once I created that model, I'm going to actually validate it out in the field using this long-term ecological research record, specifically phenocams. So this is the range of red oak, and these are phenocams, which take pictures of the canopy every 30 minutes so you can extract phenological information over very long timescales. I've gone out and I've actually sequenced all the red oak individuals growing in these phenocams using low coverage whole genome sequencing. And so I'm now working on putting all that data together, and I would love to chat with all of you about it. So yeah, if you have any questions, happy to chat now or anytime during the rest of the week.
How many,
Yeah.
In each of the sites, I couldn't see how many sites.
Yes.
It looks like 10. How many individuals are you doing whole genome sequencing on, and how did you pick this individual?
So I've gone out and you can actually identify individual red oaks growing in the phenocam viewshed. So unfortunately, I only have about probably 12 to 15 per site. So I'm trying to figure out ways of using maybe the pheno network to sort of up that number. But the goal right now is to create this model, make predictions, and then actually check if that matches sort of what it looks like across the range of red oak. Yeah, that's sort of where I'm at right now with that part.
In some controversy, shown that the maternal environment time of seed formation
Yep.
affects the phenology of the offspring. So do you think that happens in
People think so. I tried to work at the Arnold Arboretum basically because it's like a common garden. So I'm hoping that because they're in a common environment, that might help, but it's definitely a factor that may contribute.
Thank you.
Yeah.